My name is Jong Lee, and I'm a postdoc in the Mendel Lab at UT Southwestern Medical Center. I'd like to sincerely thank the Oligonucleotide Therapeutics Society for selecting our paper as one of the papers of the year. In the next 10 minutes, I will present our work described in this paper, antisense-mediated transcript knockdown triggers premature transcription termination. Long non-coding RNAs, or IncRNAs for short, are a class of non-coding transcript that have a length greater than 200 nucleotides and lack protein coding potential. IncRNAs are often classified into those that function in cis to regulate local gene expression, and those that leave the site of the transcription and perform various functions in trans, such as molecular decoy, or directly regulating target molecules in the nucleus or cytoplasm. When we consider cis-acting IncRNAs, the mere production of the transcript is not sufficient to infer a functional role for RNA itself. Rather, at least 3 possible mechanisms must be considered. First, DNA elements within the IncRNA locus regulate local gene expression, but IncRNA transcription is not required. Second, transcription of the IncRNA locus is required for regulatory activity, but the transcribed RNA itself is not functional. Or the IncRNA molecule performs an RNA-mediated regulatory function. Establishing the transcription of a IncRNA locus is necessary for its function is very straightforward and can be accomplished by introducing sequence elements that terminate transcription using genome editing technique. To establish that the RNA molecule itself is functional, the most frequently used technique is antisense oligo-mediated knockdown, which triggers RNase H-mediated cleavage of RNA. This method is very effective for IncRNA knockdown, in part due to the activity of RNase H in the nucleus, where many IncRNAs are localized. However, we realized that it was not clear if ASOs influence transcription, which would confound the interpretation of this widely used experimental approach. To understand why ASOs may interfere with the transcription, we first have to consider the canonical mechanism of transcription termination. This slide shows the torpedo mechanism of eukaryotic transcription termination. When nascent transcripts are clipped by polyadenylation machinery, the uncapped RNA that remains associated with the elongating RNA polymerase II provide an entry point for the XRN2. XRN2 then degrades the nascent transcript until it catches up with the pol II, resulting in dissociation of the transcription complex. Similarly, we propose a model in which ASO-mediated cleavage of nascent RNAs would produce an uncapped nascent RNA, which could induce the premature transcription termination by activating torpedo mechanism. Given that this could impact the interpretation of data using ASOs and possibly the therapeutic application of ASOs, we decided to investigate this hypothesis. To test if ASOs trigger degradation of nascent transcript, we perform a nuclear run-on assay in the presence of ribonucleotide precursors, including BR-UTP, allowing the specific recovery and quantification of BRU-labeled nascent transcript. Shown here are the results from analysis of 3 IncRNA, NORAD, which is the cytoplasmic, MALAT1, which is the nuclear, and hot air, which has introns. Our result from nuclear run-on experiments shows that introduction of ASO targeting these RNAs result in significant reduction in nascent transcript abundance downstream of ASO targeting site for each assayed transcript. We then performed EU pulse labeling experiment to study whether ASOs induce the degradation of nascent transcript in intact cells. Consistent with the nuclear run-on result, transfection of ASOs targeting NORAD, MALAT1, and hot air all resulted in significant reduction in chromatin-associated nascent transcript levels. Next, we tested whether ASO-mediated cleavage of nascent transcript resulted in premature transcription termination by performing chip assay to monitor occupancy of total or elongating pole 2. We found a significant reduction in both total and elongating pole 2 at 3' end of each gene body after ASO targeting. Overall, this result demonstrates that ASO-mediated cleavage does in fact trigger premature transcription termination. Our model also predicts that ASO-mediated premature transcription termination should be dependent upon XRN2. To test for XRN2 dependence, we examine nascent transcript level and pole 2 association of ASO targeted gene following XRN2 depletion. In cells depleted of XRN2, nascent transcript abundance at the 3' end of NORAD or protein coding gene PPIB was no longer significantly reduced following transfection of ASO targeting this transcript, as indicated in this highlighted box. Moreover, chip assays shows that XRN2 depletion fully restore association of pole 2 at 3' end of NORAD and PPIB gene. Therefore, these data support the model that ASO-mediated cleavage of nascent transcript induced the torpedo mechanism within targeted gene bodies. Although this result demonstrates that ASO trigger premature transcription termination, we hypothesize targeting the 3' end of gene as close as possible to the polyadenylation sequence could result in transcript knockdown while preserving pole 2 association with the gene body. To test this hypothesis, additional ASOs were designed to target the distal portion of the IncRNA NORAD. Both proximally targeting ASO1 and distally targeting ASO2 resulted in efficient and comparable knockdown of total NORAD transcript level. We then looked at the pole 2 occupancy by chip assay and found that targeting the distal portion, distal end of gene preserve pole 2 association with the distal gene body. This demonstrates that the confound effect of premature termination can be avoided by targeting ASOs to the distal segment of the gene. Our results reveal important experimental consideration when investigating RNA-mediated gene regulation in cis. To avoid the confounding effects of ASO-mediated transcript termination, we suggest targeting the distal portion of the gene of interest with the ASOs and confirming the effects on pole 2 occupancy by chip assay. In the future, it will be important to investigate orthogonal methods for introducing transcript knockdown to determine their effects on transcription termination. Additionally, the ability of ASOs to induce the transcription termination may enable new therapeutic application for ASOs. I would like to give thanks to my mentor, Dr. Mendel, for his invaluable advice and outstanding mentorship. Also, I'd like to give thanks to my lab members for their support. Lastly, I am extremely grateful to the Oligonucleotide Therapeutics Society for this great award and support. Thank you for listening to my talk.